as she said, I joined in 2019, so I still consider myself fairly new, and I've learned so much about really the industry here in the Salinas Valley, and a lot of that has shaped the research that we do on a daily basis. So first off, I just want to thank a lot of the growers and PCAs and the industry members that have been so supportive of this research. So let's see. Okay, so just a very brief outline of what I'll be covering today is really thrips and tospovirus biology. And so INSV, tomato spotted wilt virus, are two viruses you may be familiar with, whether you're involved in tomato production in the Central Valley or lettuce production here on the Central Coast, or even ornamentals. INSV has been an issue in the past as well. So we'll be talking a little bit about the symptomology, the transmission, the life cycle of the insect as well, some of the novelties of this pathosystem as well. We'll be talking about some of the research that we've done to better understand the distribution of the insect, the distribution of the virus. And then finally, I'll end with some of the organic approaches that we're taking to try to minimize the impact of both thrips and INSV in lettuce. So, all right, so here we go. Let's start from the very beginning, just a very broad slide. Just thrips as vectors for viruses affecting ag. This is a global phenomenon that we see throughout the world, both in vegetable crops and ornamental crops. There's over 2,000 species of thrips within the order of Thysanoptera and the family of Thripidae. But our primary vector that we're concerned about here, as well as in the Central Valley, is the Western flower thrips, and that's Franklin yellow oxidin talus. There's numerous other vectors as well, but this is the primary one in terms of the vector that can transmit the most number of viruses on a global scale. So a lot of different diseases, the viruses that we're talking about today, they belong in the family of tospoviridae, and that includes tomato spotted wilt virus and impatiens necrotic spot virus. And you can see that the number of crops that these viruses can infect is quite large, so over 800 plant species, and that includes both vegetables and ornamentals. And those losses are quite economical, exceeding billions of dollars on an annual basis. So I'll be diving a little bit deeper into all of this. So the biology of the insect here. A lot of you may be familiar with TSWV and tomatoes and pepper production in California here. If you're an ornamental person, coleus, a number of other flowering crops that are used for commercial purposes can also be infected. And we see that in Santa Maria and Guadalupe and even further down south in San Diego. So, but the focus of my talk, because we're in the Salinas Valley, is really lettuce. And so that's what we'll be focusing on today. I think a lot of you are familiar with this, but anytime I give a talk that has a broader audience outside of Salinas Valley, I like to just include this anyways and just talk about really the importance and the value of lettuce here in the Salinas Valley. In addition, it's quite a diverse region in terms of the vegetable crops and fruit crops that are grown here. And that's going to be really important when we start thinking about the host range and a lot of the studies that we've done to understand the distribution of thrips as well as the virus itself. So I always like to talk a little bit about some of the cultural practices, because I think that really puts into perspective some of the inherent challenges in management and the way we have to think about management moving into the future. And so I really like this picture, because what you're looking at is really this illustration of a very long growing season. So planting is actually beginning in January, last harvest occurring in November, and really the only break is this 2-week lettuce-free period in December. And so the intensity of production is quite high. And so in the background, you can see a harvest crew running through this field back here. You have another lettuce planting that is about midway through the crop cycle. And then in the foreground here, you have a field that was just planted. And so you're just seeing that germination occurring there. And so you have a lot of these overlaps and staggered plantings that are occurring throughout the Salinas Valley. So if we start to narrow down and just focus on impatiens necrotic spot virus, historically, it was actually considered a pathogen of ornamental crops, but over the past probably 10 to 20 years, it's really gained recognition for being a pathogen of vegetables in both Europe and North America as well. And so in the Salinas Valley, 2006 was when it was first documented in lettuce. And then from there until about 2012, we've seen some minor to severe isolated outbreaks. I started in 2019, so really I started right at the beginning of some of the epidemics that have the Salinas Valley has experienced. And so since 2019, we've seen quite severe outbreaks, up to 100% crop losses. And the estimate from 2022 was over $150 million lost. And that was through a lot of the monitoring and reporting systems that were generated by the Grower Shipper Association here for Central California. And they've been absolutely critical in terms of helping us document the impact of the disease, which has helped really, get better recognition on a national level to try to do some bigger research projects and really highlight the importance and the value of the crop in this area. So let's see. So I'm just going to run through some symptomology because I think the pictures really speak for themselves. But you're looking at a romaine lettuce plant right here. And so the name impatiens necrotic spot virus is because it was first described in impatiens plants. But the necrosis comes from the symptoms that are experienced in the leaf tissues here. And so in this case of romaine, you're seeing an early, very minor symptoms. And so just that yellowing that's occurring on the leaves. On the right side, you see advanced development of the symptomology. And so very severe necrosis. And this can also lead to very severe stunting of the plant, particularly in the heart, and then ultimately affecting the leaf morphology as well. And so this is just an overhead picture of a field of lettuce. And so what you're looking at, this plant right here, at least it's asymptomatic. It could be infected, at least it's not showing any symptoms at the time. But you have a wide array of different symptoms that we're looking at here. As I mentioned, the stunting of the growth here, the very strong necrosis that's occurring in the leaves, and then also some of the twisting of the leaves where you see it, it's really not forming that tight head like this plant is about to start shortly in the next couple of weeks. So the infection can occur very early. So this is a crop that has not been thinned yet. So it's a direct seeded crop, hasn't been thinned, and you're already seeing very strong necrosis that's occurring at this very young stage. So there's a lot of different symptoms that can show up in lettuce. And part of that is because there's such a wide variety of varieties that are grown commercially. And so you just think about a lot of the different underlying genetics in these different varieties that might lead to different physiologies of the plant. And I think that ultimately affects how the symptomology shows up in this plant as well. So in contrast to some of those romaine pictures I was showing you, here you're seeing necrosis that's primarily restricted to the bottom of the plant in the midribs. And so it's been, it's one of the big educational aspects that we've really tried to emphasize is just scouting and recognition of the virus as well. And part of that is just through, pictures like this and just building more information as we get it. So this is a head lettuce plant. And so it's been cut and it's the same phenomenon as that. The necrosis, you can actually see a lot of the spots that are occurring here in the midribs at the bottom of the plant too. And this picture, as well as this picture, the reason why these are particularly concerning is because it makes it very difficult for a harvest crew. So if you look at this picture, the plants look really good, but then once you have issues in the bottom, it can be very difficult to assess really how much of that product will make it into the box. There's also questions of stability and shipment, and we don't really have those answers yet, whether these early infections will affect the shelf life of the crop. So a lot of research that we're really interested in, but don't have the answers for it yet. So same plant. And then this is just another picture of very early infections that you're seeing on the leaf. If you look at this plant here on the left, this is actually that same plant. You could see the heart that's starting to stunt in its growth as well. So, and then really this virus is transmitted by its insect vector, which is the Western flower thrips, which we introduced in one of our very first slides. And so they're quite small insects. They're ubiquitous. You can see this very young lettuce plant that has numerous adults, and so each one of these are adults. There's 2 of them right here, and they're quite small, and so when you start thinking about management, it becomes very challenging. So and I'll just point out, this is very indicative of damage, and so you see the silvering of the leaf, the scarring that's occurring, and that can have an impact on the product as well. So if we talk about just the life cycle of thrips, we mentioned that there's, they're small nature, they're very cryptic, and there's a lot of some big chemical limitations in terms of how we manage it. And so if you think about production across the entire Salinas Valley, you have, there's a very rough estimates, 80% conventional, 20% organic, with just fundamental differences in how they can manage a field. How do you generate a consistent IPM program throughout all lettuce that's grown in the valley? And that's very difficult. And so we're trying to address things, looking at the conventional side, looking at the organic side, but also things that can be applied both through cultural methods as well. And so we'll talk a little bit about that today. So the management side, I mentioned the host range for both the virus and the insect is quite large. In the diverse ecosystem that we have here, it becomes difficult. So let's see. So some unique features about thrips and these tospoviruses that they transmit, they're quite different in compared to other insect vectors. So we think about aphids and leafhoppers and whiteflies. And really the mode of transmission of how the viruses are transmitted by the insect is quite different. And so I'll just run through this, and then I actually have some pictures to help illustrate the transmission as well. But it's the adult form. So the male on the left and the female, they both transmit the virus. It's not, once they have the virus, it does not transfer down into the next generation. And so they have to reacquire it each generation. But what's tricky here is that in order for the adult to transmit the virus, they have to acquire it at this very first instar stage. And so this instar has to feed on an infected plant, which essentially means that the egg from the previous adult has to be laid into an infected plant. You get the L1 that emerges from it, acquires the virus, and then they continue their life cycle into adulthood where transmission occurs. So keep that in mind. I'm going to come back to it and just show you in an illustration, and I think it'll really help understand this slide a bit more. But okay, so here we go. So we have multiple scenarios. So we have an uninfected plant, an INSV infected plant, and we have really another uninfected plant, but what is the status of this plant? And so in scenario 1, these are kind of no-brainers, right? So if you have an adult that feeds on an uninfected plant, you don't get transmission to another plant, right? If you get reproduction on that crop, again, you don't get transmission. What's tricky is that if you get an adult that feeds on an infected plant, you still don't get transmission to this plant. The only way that this plant can be infected is, again, if they lay their eggs in that plant you get the L1 that emerges, and they feed off of that infected plant. So they acquire the virus, and then they complete their life cycle into adulthood. And that is the generation that can transmit the virus. Okay. And so if you look at it from a field perspective, you can have what we call primary infections, where you have these adults that are coming into the field, introducing the virus to the field. And then you may also get secondary infection, where you get active reproduction. And so again, when we start thinking about management, a lot of us are just primarily focused on the adults, but we were in a meeting yesterday, several other meetings, thinking about how to break this cycle. And really, are there ways that we can manage this life cycle as well, especially on infected tissues? And so I always present this slide, and I kind of call this my nightmare slide, if you will. And what you're looking at is an infected lettuce plant. And so you're looking at a plant that is infected with INSV, but you also see the larvae that are on those infected leaves. And what that means to me is that you have larvae that are acquiring the virus, which means that once they complete their life cycle into adulthood, they can then transmit the virus. And so that, to me, this is the most riskiest situation that you have within a crop. So this was just some of the work. Some of you may know Alejandro Del Pozo, who is one of the former farm advisors here in Monterey County. He and I worked together on this in 2019. And really, we just documented the severity and the spread of this disease within a single field. So this is about a 10-acre field. And what you're looking at the top is going to be the severity. So how strong of a symptom you see in the plants. And you'll see it as it gets worse, it gets dark red. On the bottom right, we did some spatial analysis, and what you're looking at is where is the virus actually aggregating within the field. And so where you see the pink, that means that's where the virus is aggregating. And so as we progress, so the planting date we call week 1, the harvest period was about 10 weeks, 9 weeks later. And as we progress through the crop cycle, we're evaluating over 1,000 individual plants every single week, and see a rapid development of infection, the severity, but then also the disease is strongly aggregating in the bottom right corner where all these pink this pink area is. And so we also did this for thrips. So we wanted to look at how the vector is moving within these fields as well. And so I'm going to keep the bottom here. And so what you're looking at is where the disease is accumulating within the field. And then on the top, you'll see very similar pictures on where the thrips are accumulating. So again, focus on the pink area. And so what we saw for week 4, 5, 6, all the way up through week 9, is that you don't see that same effect, right? You don't see that most of the thrips are coming in from the bottom right corner of that field. It's actually widely distributed. You have it coming in from the top side, which is the north in this case, in different areas of the field, infestations occurring in all directions. And really the take-home message here is that thrips distribution does not always equal INSV distribution. So again, another challenge for just thinking about scouting, both for the disease and also scouting for the insect. The reason why we had so much disease here was that we actually had another lettuce planting that was adjacent to it. And so the insects that were coming, the thrips that were coming out of that field, a higher percentage of them was carrying the virus. And that's why you see that strong gradient effect. But simultaneously, you have very strong winds in the Salinas Valley, a lot of other crop activities occurring that is driving infestation occurring from other areas of the field, but they're not necessarily carrying the virus. And so that's been kind of a big message that we've talked about as well. And that's really just fundamental vector biology as well, in terms of how to understand the epidemiology of it. So when we look at the disease in orange, so you look at this, the incidence of INSV in this field over a 9-week period, and then we also quantified the number of thrips per lettuce plant every single week as well, and you see that increase also. And so One of the things that we're starting to explore is how to improve the coverage of whether it's your organic materials or your conventional materials, because the morphology of a lettuce plant drastically changes over the crop cycle. And what we see as you go through plant maturity is that those insects are really at the bottom of the plant. And so how do we develop better management solutions while adapting to really the structure and the morphology of this crop? So we run a thrips monitoring program here. Generally, what we see is that thrips increase throughout the Salinas Valley throughout the entire year, and that generally correlates with INSV as well. So you have higher vector populations, you have more disease incidence as well. So how did we get to this state where we currently are in the industry? And so this is just kind of theoretical at this point, but we just pulled some weather data from one of the CMEs locations for the past 20 years. And you look at the minimum temperature development for thrips, it's this red line right here. And then you look at really the average minimum air temperature in blue, and we see that line increase above the minimum temperature for thrips development. And so that would suggest that you are actually getting active development of the insect even in the winter time. And that's what some of our data is actually showing here within the past 3 years. But what that means is that we don't have these hard winters anymore where you really have a break in the vector population. You have active populations throughout the year. And so we think that that could be part of the reason why we have so many challenges with managing the insect and the virus today. So let's see. INSV host range, thrips host range as well. So we're going to talk a little bit about dispersal. So I mentioned the strong winds, and what we see here is that thrips are not great flyers on their own, right? They're actually poor flyers. And if you look at their wing structure, they're not true wings, right? They have these little bristles in the back, and they rely heavily on the wind for dispersal. And because they're small, it's hard to estimate distance. But some of the things that we did last year was we put sticky cards up on telephone poles. It's kind of a fun project, and we just wanted to see how high in the wind column we would actually capture some of these thrips. And it was surprising that we were catching them, even up to 8 or 9 feet and fairly equally distributed. How the distance is hard to measure, but when you start thinking about management, how do you protect yourself and your crop from an insect that has the potential to be pretty high up in the wind column? And so we're trying to think creatively about how to do that, but just raise awareness more about some of the activities that are happening beyond the farm as well. So some of the surveys that we just started this past year, I have an awesome undergrad who's been on this project, and we're just surveying a lot of both crops, but also non-crops, to start to build these indices on where are our thrips. And so most of these crops, these were sampled during the spring and the summertime. What I'll say is that just because it's a thrips host doesn't necessarily mean it's a virus host. And so there are some additional separate studies that I'll talk about that addresses some of that as well. But in general, we just wanted to see, start to get an idea of where are the thrips within the landscape. And this is all in the Salinas Valley. So we have non-lettuce crops that can be great hosts for thrips, but we also have non-crops that can be really good hosts for thrips. One of the things I'll point out is sweet alyssum. We were a little bit surprised that sweet alyssum didn't have a lot of thrips, and that's actually good news because it's often intercropped in some of our organic systems as insectary plants. So this was actually looking at hosts for the virus itself, and so we really wanted to see where was the virus in these non-lettuce crops throughout the year. And so we've set up 4 different sampling periods, this last one being the most important during the lettuce-free period. We did a variety of detection methods, both serological and PCR. We've sampled probably close to 4,000 plants at this point, covering 73 different species of weeds and vegetable crops and native plants. what we came up with was really a top 10 list of hosts that can serve as a reservoir for INSV. And this was working closely with Richard Smith, who helped with the sampling, as well as the identification of these different species. And so these are all weedy species that we've characterized as being pretty good hosts for the virus. And what's most important about this is that their abundance is quite prolific, right? We see them year round, even in the winter time. And I think that's, sort of unique for a lot of California, especially on the Central Coast, where we have where we've had a lot of wet winters this past year, but also we don't have those strong heats that helps with cycling out some of these weeds during the summertime. They're really prevalent year round in our cropping system here. So, and really the important piece here is that if you look at here, this lettuce-free period where we have no lettuce in the Salinas Valley, we found that within these top 10 hosts, we found a high percentage of them tested positive for the virus. And so what this means is that a lot of these weeds are actually serving as really the green bridge for the virus between subsequent lettuce seasons. And so one of the things that we've really focused on is, trying to take these strategic management strategies that both balance concerns environmentally, right? We don't want to necessarily promote a scorched earth policy, but how do we focus on minimizing the impact to our growers, but also balance really the interest of being good stewards of our own land and the environment. So, and we've worked with Monterey County on this quite a bit. So this was I just threw this in here. This was an article we published with Richard Smith a few days ago. And this is really non-INSV host plants for habitat planting. This is some of the numbers here are pretty small, but we're continuing to do this work. And this was part of that whole study where we were doing the weed surveys. And so we're continuing that, but we're trying to make recommendations as well on things that are generally really good plants that could be grown in organic systems to improve the habitats and the environment as well. So, okay. And then the last thing that I'm just going to finish up on are just two things that we're working on in terms of trying to improve some of the organic solutions for our industry here. And so the first thing is immune priming. So overall, how do we boost plant health? And so this is work that we've started with Carrie Mock. She's a professor at UC Riverside. It's a 2.5-year project. We've just completed about 1.5 years actually at this point. Some greenhouse studies. We just finished a field trial. But we're looking at a lot of these different priming agents to see if it could manage or suppress the severity of INSV infection. Carrie's done a lot of this work in melons, and she's had a lot of support from the California Melon Board, and really through the lens of managing whitefly transmitted viruses. And she's had quite a bit of success there. And so when we started this project in 2021, it was, can we adapt these technologies and see if we can apply it to lettuce? And so what you're looking at are some trials that were completed in the greenhouse. And it seems like there might be some really promising effects in terms of some of these products in minimizing some of the severity that's caused by the virus. So we're also looking at overall plant metabolites and how some of these priming agents might affect plant health as well. So, and then the last thing I'm going to talk about quickly, I'm not going to spend too much time here, but this is a project that I'm collaborating with, and Ian Grettenberger at UC Davis, who many of you may know, is leading this trial. He's a specialist for UC, and he's based out of Davis. His PhD student, Addie Abrams, is the one that's doing a lot of this work. And the idea is using these new spray technologies. So the Mantis, which is a precision sprayer, to see if we can improve some of the management of aphids and thrips in lettuce production. So won't spend really a lot of detail here. We just tested 4 products here, a Pyganic, Entrust, Nemex, and then Microtrol ESO. And they were all applied at the label rate, the per acre label max rate. And so just to give you an idea, the manual thinning stage right here, this is when we go in with the Mantis and make those applications. And so what you're doing is you're actually minimizing how much spray is being applied on a per acre basis. And so the estimated amount in reduction is that you can actually reduce it down, reduce like 90% of the insecticide that you're using, because you're using these smart technologies that are making directed applications to the plant. So we had two applications that were made here. We sampled it before and after the applications, and we also did an INSV evaluation too. For the Thrips studies, really nothing too much to brag about here, other than we saw a little bit of a reduction caused by the Mantis using the microtrol product here. We saw a general reduction, but it wasn't statistically significant for entrust. And this is comparing both the broadcast application as well as the precision sprayer. So we didn't see any differences in INSV incidence, likely because we didn't have very high pressure. So you look at the overall incidence, we're only at about 1% here. So we're going to repeat some of these studies this year in a higher pressure area. So, and then the last thing for aphids, when we looked at these products against aphid management, we saw two significant differences there in these two products using the precision sprayer versus the broadcast application. So we're also doing some studies that I'm not going to talk about today, looking at augmentative biocontrol, so releasing beneficials. Again, that's being led by Ian Grettenberger, and I'm sure we'll connect with you all in the future, and we'll have a little bit more data to talk about that next time. So anyways, this is our working model, and this is my last slide here, is just how do we think about INSV management versus Thrips management, and just kind of bring it all together. And so it's a very complicated system. Anytime you're dealing with a vector, it is complex, but I think we have to continue thinking about IPM through the lens of your environment, right? And so every one of you is going to be in a very different growing environment, and the IPM model is going to have to shift depending on that. So, and that's vegetables, ornamentals, fruits, whatever it may be. So anyways, this is for lettuce in the Salinas Valley. So anyways, that is all I have for you all. I want to thank you all for listening, and hope you all had a good lunch, but reach out to me if you have any questions.